brief presentation of myself. I am a biotechnologist, and I work in the laboratory of preclinical molecular imaging of the CNR. It is located within the Sara Institute with a formal agreement, and this agreement covers also the collaboration with the University of Milano Bicocca. And through this collaboration, we have access to two different radiochemistry for the labeling of different PET and SPECT probes, and for the application in small animal models of oncology, neurodegeneration, and inflammation. And we have state-of-the-art devices for in vivo preclinical imaging, as CT, PET, SPECT, and also optical imaging. And we have access also to the MRI, preclinical MRI. And just to show a typical experimental workflow that provides animal model preparation and image acquisition and data analysis. And for the analysis, we are available a few mouse and rat templates, but here you can show an example. But usually these templates are general and sometimes not so useful in understanding disease behavior because of a small partition of the areas and also no lateralization. And also in the studies, we can observe low standardization in disease definition and analysis, and also often no standardization in space coordinates. And we have no reference because of different instrumentation and analysis, and most of the PET studies on brain need to have MRI acquisition to improve signal localization. So we need a simple and automated process for image normalization of the PET brain image and for the analysis in mouse. And with this aim, we try to build a system for the definition of common brain volume space for the mouse brain. And we exploit the Allen Brain Atlas, the brain atlas of mouse deriving from the Allen Institute. And we try to define a volume of interest based on this common atlas. And we built MRI, PET, and CT image template starting from this volume. And we set a sample and automatic script for the easy use of the templates within the SPM language. And also we do CT spatial normalization. And here you can see the website of the Allen Brain Atlas, and on the right you have an example of the template pipeline that we have used for the normalization of the images. And the template was built by using healthy animals that was acquired with both FDG PET and CT. And a part of them will also has been also acquired with high resolution 3D MRI imaging. And a set of scripts has been done. And this is the screenshot of the optimized batch provided for the building of 27 regions within all the brain. As you can see, there are a very small regions, but these regions are in agreement with the spatial resolution of the PET. And here you can see the optimized batch for the preclinical mouse imaging. So some template is built using value of that are comparable of the scale of the brain mouse. A key aspect when we built our package is to make it available in the larger scientific community. Therefore, it is freely available for the download by the NITRC website, and until now we have more than 200 downloaded. And we published all the procedure in this draft. So we validated the toolbox in different Parkinson's disease model mouse obtained by injecting neurotoxin or using transgenic mouse model. The first model used is obtained by the MPTP injection within a week. So we obtain a subacute model in which we observed the decrease of the dopamine transporter using TH immunostaining, and also an increase of the IBA1 immunostaining, so the increase of neuroinflammation. And so the imaging protocol consists of an acquisition of the specific radioligand for neuroinflammation, the VC701, that is for preclinical use. So we have the acquisition at baseline, we have the treatment within 7 days of MPTP, and then we have the imaging of the same animal after the end of the treatment. This is the protocols and the results of this study. You can see the protocols for CT and PET acquisition and reconstruction. And here you have a table in which we apply our template of 27 regions in comparison with the classical template of PMOD. And as you can see, we are able to identify 7 additional regions in which we observe a significant increase of the uptake of our radiotracer specific for neuroinflammation. And here you can see also a map in which you observe the statistical increase of the signal within the regions involved in the disease. So you have signal in different part of frontal cortex, within striatum, and also in other part of cortex, and also in thalamus. And we observe also a correlation between the area involved in the disease by applying the template in the tool of the Allen Brain Atlas. So we apply this tool in a second model of Parkinson's disease. This is a more progressive model obtained by the injection of MPTP within 5 weeks. So the imaging protocol consists in the acquisition of 2 different radioligands. One is specific for dopamine transporter, and one for is the FDG that measure glucose metabolism and also connectivity. And we done the acquisition at baseline and also at a different time point after the administration of the MPTP. And this model is characterized again by a decrease in dopamine content. And by using PET and our tool, we are able to observe a different behavior in the dorsal and the ventral part of the striatum. And this decrease in dopamine content correlated also with the data measured by immunohistochemistry. And also with FDG, we observe a decrease of the glucose metabolism within dorsal and ventral part of the striatum. And we are able, by applying our template, also to do connectivity, and we measure the difference in connectivity between over the time between subcortical and cortical areas. So we applied our tool in a third model of Parkinson's disease, is a transgenic mouse model obtained by the mutated form of alpha-synuclein. It contains human mutated form of alpha-synuclein, and we study this animal model at different time points just to observe the behavior in a very preclinical phase of the disease in order to identify very early marker of the disease. We performed also MRI using high resolution sequences just to measure the volume of the whole brain, and we observe in this case that there is a significant difference in the volume of the mutated animal compared to the control. And by using our radioligand for the dopamine transporter, we observe a difference in the ventral part of the striatum at 8 months after 8 months of age. And our tool can be also used for an integration with radiomic analysis for the extraction of image features. These features are related to intensity and also and also size and shape of the images, and we obtained also a variation of this the region involved in the disease at different time point of the age of the animals. So in conclusion, we can say that we created this toolbox for mouse brain PET image analysis using a common coordinate deriving from the Allen Brain Atlas that allow us to define 27 lateralized regions and for the automatic extraction of the signal without having MRI, because this tool is based on the CT. So we improve signal quantification using the toolbox, and and allow image normalization for different analysis purposes, as for example radiomics and also connectivity of the brain regions. And we applied this tool to different Parkinson's disease model and to different PET radioligands, improving identification of the areas involved in the disease, and data correlated with also ex vivo immunostaining. So this tool could be potentially applied to any brain disorders and PET radiotracers. These are the people that are involved in this study, and I thank you for the attention.